Enzymes are proteins that help speed up chemical reactions in our bodies. Enzymes are essential for digestion, liver function, and much more. Too much or too little of certain enzyme can cause health problems. Enzymes in our body can also help healthcare providers check for injuries and diseases. What are the factors that affect of enzymes activity? First is pH. Enzymes are sensitive to acidity and alkalinity. They don't work properly if an environment is too acidic or basic. For example, an enzyme in the stomach called pepsin breaks down proteins. If the stomach doesn't have enough acid, pepsin can function optimally.
Temperature. Enzymes work best when your body temperature is normal, about 98.6 degrees Fahrenheit or 37 degrees Celsius. As temperature increases, enzyme reaction increase. But if the temperature gets too high, the enzyme stop working. That's why a high fever can disrupt bodily functions. Enzyme assay. An enzyme assay is a lab technique to measure enzyme activity by monitoring the rate of a specific chemical reaction catalyzed by the enzyme. It helps in studying enzyme kinetics, determining enzyme concentrations, and evaluating enzyme purity and stability.
The most produced bubbles is the test tube pore. This is because acetic acid provides a more acidic environment compared to the other substance in the test tubes. Catalysis, an enzyme responsible for breaking down hydrogen peroxide into water and oxygen, functions optimally in a slightly acidic environment. Therefore, the presence of acetic acid in the test tube pores creates conditions that enhance the activity of catalysts, resulting in more bubbles being produced as oxygen gas and released during the reaction. Effect of enzymatic concentration and enzyme activity. The effect of enzymatic concentration and enzyme activity is typically described by the concept of enzyme kinetics. Generally, increasing the concentration of enzymes in a reaction increases the rate of reaction. Upon to a certain point, this is because more enzyme molecules are available to catalyze the conversion of substrate molecules into products. However, once all the substrate molecules are being acted upon by the enzyme molecules, or enzyme saturation further increases and enzyme concentration may not significantly increase the reaction rate. In conclusion, the experiments conducted aim to assess the impact of catalase concentration on the reaction rate using various samples such as potato or sweet potato, yeast, chicken liver, and young leaves. The results consistently demonstrated a correlation between increased catalase concentration and higher production of bubbles in the active of an elevated reaction rate. This aligns with the general understanding that as enzyme concentration rise, they are more active.